of the main differences, I think, one, it's a lot harder to try to look at this in humans. Part of our study and part of other studies within this similar field, it's really hard to find HIV in the circulating monocytes in patients. So essentially, to do a similar type of study, what you really need access to are patient tissue samples. So this is a much harder to come across, and it's a much more invasive process if you want to look at humans instead. What this demonstrates to us is that we can't discount macrophages as a viable source of virus. So especially considering one of the viruses that replicated well was directly derived from a patient. So we are showing that this is a real world phenomenon. So while it may not apply to all patients, it may emphasize to us that better screening to figure out which patients may be harboring viruses that are capable of infecting macrophages and seeing, are they having kind of different effects from the virus in a different course of disease because they have an additional cell type that can also replicate the virus. So what our preliminary experiments have shown so far with regard to antiretroviral treatment of infected macrophages, what we've found in the mice that we've looked at so far is that the current regimens we have are actually quite effective at suppressing the virus in the macrophages. So in particular, we've been looking at the regimen containing intracitabine, tenofovir, and raltegravir. And so far, we've seen very promising results that the existing regimens will likely be very effective against macrophages as well. So we are certainly excited to be able to test antiretroviral therapy in these mice. So we've already evaluated antiretroviral therapy in our humanized BLT, which are bone marrow, liver, thymus mice, which have all the components of the immune system. And we've also tested this particular antiretroviral therapy regimen in our humanized T-cell only mice. So the experiments are actually currently underway to evaluate this same regimen in the humanized myeloid only mice. So those experiments are ongoing, and we hope to have a publication together in the near future. Our main goal long-term with this model is in terms of HIV cure. So important to figuring out a cure for HIV will be to also figure out what are all the players involved. So what we really want to look at is whether or not macrophages represent a viral reservoir in patients that are well suppressed on therapy. So our big end goal would be to determine whether or not these cells are a reservoir for virus, and then in which we can start to look at macrophage specific targeting towards a cure.